Hi, I'm Bill Rapisi, Executive Director of Lymphatic Education and Research Network, and I just want to take this opportunity to thank all of you for joining us today for our Symposium series. I also want to take this opportunity to thank all of you who become members of LE&RN and help support this series so that we can continue bringing this kind of groundbreaking information to you in your homes and offices. I would also suggest for all of you who have not become members of LE&RN, $5 a month, you can become a member of LE&RN and help support this programming and the research that we do. Thanks very much for joining us, and I look forward to having you become a member.
Well, thanks for joining me online and in the room as I talk a little bit about the work that my lab has been doing in lymphatics, and particularly with an interest in how mechanics is involved in both lymphatic function and lymphedema, which I know is the interest of the people in the audience today. Real quickly, just so I make sure I get to it, this is a disclosure. We have a company that we've launched out of Georgia Tech in lymphatic diagnostics called Lymphatech, and I own equity. I have a financial interest in that. Okay, so the nice thing about talking to this group is I don't have to introduce the lymphatic system in much detail, but just to provide some context and a framework for our discussions, when I refer to the lymphatic system, I'm mainly referring to this system of vessels that exist throughout your body, often running in parallel to the circulation, that transports fluid from the interstitium back into your venous circulation. In addition to that, it provides a network for immune cell trafficking to lymph nodes and also all fat transport from the intestine. So when we think about the lymphatic system and its functions, and we think about lymphatic anatomy, we generally divide it into two very broad categories. So we have the initial lymphatic capillaries. These they don't have any smooth muscle or basement membrane. They often are described as sort of leaky, so that big molecules can get in them. And those merge into the collecting lymphatic vessels, which we often refer to as Lymphangion's, and these are the things that pump to move fluid up gravitational loads and up the pressure gradient that exists to return it to central venous pressure. And now the lymphatics importance is probably most obvious when we show one of these images of lymphatic disease known as lymphedema, in which the lymphatics fail, and then over time you get this accumulation of fluid which turns into tissue, fibrotic tissue, fat, etc., and the limb grows. What we really don't understand, I think, at a very detailed level, is how the lymphatics themselves are involved in this process and how lymphatic failure might be driving it. I mean, we certainly know that things like surgery or tissue removal or a blockage can help start the problem, but it's how it continues and progresses is less clear. Now, I often get asked, why is an engineer studying the lymphatic system or lymphatic physiology? And so perhaps one analogy I could make is to this sort of complicated system of pipes. And you could look at this and see, I could see how that's an engineering problem, right? Where I have pipes with varying resistances and I've got to move fluid from one direction to another. And so maybe we could think of lymphatics like a blockage of pipes. Or for those of us in Atlanta, maybe an analogy of lymphatic disease that comes close to home. A year or so ago, we got like 2 inches of snow, and this is what happened to all of our highways. And so as we have this blockage in sort of the main collector freeway that runs through the middle of downtown, no one could leave the city and all the surface roads back up. But at the end of the day, to resolve this traffic problem, the people have to get back on the highway so that the 6 million of us that commute into Atlanta can get back to our homes. And so similarly, while I can certainly have fluid drive through the lymphatic capillaries in sort of these pathological or very stressful situations, at the end of the day, they need to merge into collecting lymphatics. And so we could certainly think of these systems as an engineer might think of them. And then, of course, ultimately we think about the pump. So this is the motor unit that really drives lymph flow. Obviously, you can look at this and think, I could see how this is a system or a vessel in which engineering principles might drive its performance. And so it contracts There's these valves that sort of close to make the flow go only in one direction, and I need all these things working correctly in order for fluid to be moved from one point to another point when the pressure at the point it's trying to move it towards is greater, which would be the case if I'm moving, for example, fluid up my leg. Now, lymphatics, mechanics is not only important in the day-to-day operation, but we can think of mechanics as being important into what happens to a biology over time. And so as this sort of cartoon illustrates, an obvious example, if you want to gain muscle mass, right, you expose your muscle to continuous load. And over time, as you add more load to your muscle grows and expands. And this happens throughout your body, throughout life and development. It's a reason one of the main concerns of sending people into space for months at a time is they're not standing up anymore, and so all their muscles and bones decrease because they don't need to support the load. And your body really only wants to expend energy to create mass when it needs to. So we can certainly think about it, and then we can also think about it in the context of disease. And I bring this example up mainly because it's close to home. So this paper came out in the 80s, and it was actually an aerospace engineer here at Georgia Tech who met with some surgeons from the University of Chicago and collaborated. And what they observed was, in this disease known as atherosclerosis, where you get these plaques that form in the inner vessel wall, what they found was that the mechanics themselves, the mechanics of the fluid, could explain where the disease occurs. And so, without getting into too much detail, in regions on the vessel where the force that the fluid exerts on the wall of the vessel, where that was sort of abnormal and oscillated back and forth, that was the place in where disease occurs. So this paper has been cited over 2,000 times. I would say this and papers like this that came around that time really created this field in which we begin to think about how mechanics might drive disease. Although I would say we really haven't even begun to think about this in the context of lymphatics. But if we look at the clinical data that is out there in regards to what lymphatics look like as the course of disease progresses, there certainly seems to be some evidence that these lymphatics remodel. Certainly there's been a lot of work on things like growth factors. So there's a growth factor called VEGF-C, which helps lymphatics grow. So certainly we have some understanding of how chemicals might be released and might enhance lymphatic growth. But what about the underlying mechanics? And during the disease, as the mechanical environment changes, how do these vessels grow and repair? And how does that in turn affect their ability to, in the end of the day, do their job, right? Move fluid from point A to point B. So this brings me to my sort of two central research questions for this talk that our lab sort of thinks about. And so one is, in the context of sort of normal lymphatic physiology and pumpings, how do mechanics interact with this system? And how does it respond? And then now once we understand that, when we start thinking about it in the context of failure or disease, how do mechanics drive that process and play a role in these pathologies that are being described in animal models and in clinical biopsies and specimens? So we're engineers, and so I like to think of this sort of cycle that we continuously iterate around. And when we try to think about improving, at the end of the day, right, we want to improve the access to patients of therapeutic options. And so there's a question regarding to, what in the end of the day, what do I need to measure? What is causing the lymphatic failure? And that's really a biological question. Maybe it's I need to measure the protein expression of a particular protein, or maybe it's the circulating level of a particular cytokine, or maybe it's just the pumping of a vessel, or maybe it's the number of vessels. We don't know, but there's a biological question of underlying in the biology, what is driving this lymphatic failure, and what do I need to measure? And then oftentimes we may want to measure something that current technology does not allow us to measure. And so then we need to think, well, how do I measure that? So, and I'll give you some examples of that. And these really feed on each other, because as we develop new ways to measure lymphatic function, then we gain new biological insight into what's causing its failure. So I'm going to start with sort of the tools aspect in our lab. And so I'm going to give you a couple of examples. So obviously, since we're interested in mechanics, one of the things we wanted to be able to do is take these lymphatic vessels and do very precise mechanical testing on them to figure out, how well do they perform, right? We could think of it as a pump, and I really want to understand the characteristics of its pump. So what are the maximum forces it can produce, how much energy does it require, et cetera, et cetera. And so really building off of all the isolated vessel work of De Zavea and Geshev and Davis and various people that really have pioneered this work, we developed a system where we could take these vessels and expose them to really you could think of it as any force we want to, any physiologic force. Now this is a little bit of a complicated diagram. I'd like to compare it to maybe something like cruise control in your car. So in your car, you get on the highway, you could set your car to go 65 miles an hour, you set it, and then you don't do anything, right? And your car, puts down the gas or takes off the gas and adjusts as it needs to maintain that velocity. We call that feedback control. We do something here where we put this vessel on and we say, okay, here's the pressure and here's the flow I want this vessel to experience, right? Because we've taken the vessel down to the bottom and we've dissected it out of an animal. So it's not in its normal environment, but we now want to recreate that normal environment. So we put this vessel on there and we use this process known as feedback control to basically prescribe the flow and the pressure on the vessel that we want. And in doing so, we can make some interesting measurements and observe some unique observations. And I'll just give you two such examples. So in this case here at the very top, you have traces of the diameter of this vessel over time. And in the second column, this is basically flow through the vessel. And so what we're doing in this experiment is when we start the experiment, there's no flow being pushed through the vessel, but then we gradually start to push fluid through the vessel. And then the third column, the third line there is, or row, is the pressure. But we do this while we hold the pressure constant. And what this allows us to do is, if you notice the diameter tracing, you'll notice the diameter stops contracting. And so this was shown originally by Toy Geshev in 2002 with the De Zavea's group, that lymphatics will stop pumping when there's already flow present. If you think of it, again, if we think the body wants to minimize energy expenditure, if there's already fluid moving through the lymphatic system, I don't need to pump. I just need to open up and allow that fluid to pass. And so what we were able to do here is we can figure out and provide a number, if you will, of exactly when, at what flow value, or what we call shear stress value, does that start to happen. And this feedback control system allows us to do that. The second thing we can do is we had an interesting question that, so when we think about lymphatics and lymphangions, we know these pumps are sort of lined up one after another in series. And so one of our thoughts was, could flow help coordinate the contraction of these vessels? So you could think of, maybe you have a vessel here, it pushes fluid out and basically expels it into this vessel. This vessel feels that fluid arrive, much like when you blow on your hand, you feel the wind on your hand. So that vessel can feel it, and as such, it can contract and use the flow itself to coordinate the contraction. At least that was sort of a hypothesis of, could this occur? And so we did this system where essentially we have the vessel contracting. In the middle, we have no flow through the vessel. So again, there's no flow, so the vessel is pumping sort of at this inherent, what we call a pacemaker frequency, to try to get flow moving. And then throughout this whole experiment, we're holding the pressure constant. And then what we do is at some point in time, we start pushing flow through it. So accelerating flow and then decelerating, accelerating, decelerating. Much like would occur when a lymphatic sends fluid into another lymphatic. And you notice the moment we start doing that, this lymphatic vessel starts contracting with that flow waveform, suggesting that, in fact, flow itself can coordinate lymphatic function. So you can think of these vessels as it's a very smart, mechanically controlled system of pumps in which the timing can actually be regulated and controlled by the mechanics themselves. So one of the questions now that we have, that we've observed this, is this compromised in disease? And is that something we need to be thinking about? In the vascular field, one of the clinically used measurement people often refer to as something known as flow-mediated dilation. So essentially, in your brachial artery, we can do a measurement to see if your vessel responds to flow by dilating. We can do that clinically, and we know that if it doesn't, if that's impaired, you're at severe risk for cardiovascular disease. And so we could begin to think about these kind of things occurring in the lymphatics. And is the impairment of the response to flow indicative of lymphatic failure risk. In addition to this, we also want to do, we wanted to be able to mechanically test these vessels. And so this is actually an entire Ph.D. thesis fit on one slide. But the rest of it is all math, and I figured you don't want to see that. But essentially what we were able to do is develop models, mathematical or computer models, that sort of capture the way lymphatics behave mechanically. And so this figure on the left, the circles and the little stars are experimental data. So these are measurements we make on lymphatic vessels under all sorts of different conditions. And then the lines are what the mathematical models that map up to those measurements. So now you can think of it, we have a virtual lymphatic vessel, if you will, that responds to mechanics. And then we can now run simulations, and we could have a virtual lymphedema patient, right? And we could say, let's change the mechanics and let our models adapt, and then compare that against experiments. And then the nice thing about a computer model is I can run thousands of experiments without sacrificing any animals, and it just takes a lot of computer time. And so this is something that we're very interested in, and this is with a collaboration with Dr. Rudy Gleason here at Georgia Tech. Okay, now in addition to mechanical testing of these vessels, all this work is done on vessels we've taken out. We want to be able to image lymphatics in what we say in vivo, which basically means in an animal. I mean, ultimately the goal is in humans. What we do is develop animal models of disease so that we can then test underlying mechanisms and screen for potential therapeutic approaches. So we use this approach. Near-infrared imaging was really first pioneered by Uno out in Japan and Ivo Sevic-Maraka in Houston. And so it essentially involves the injection of a near-infrared tracer in the dermal layer of a particular molecular weight, and that gets taken up into lymphatics. And because the light is near-infrared, it can penetrate through the skin decently far, and I can see all the superficial lymphatics. And here's just some examples. In this case here on the right, we have a lymphatic vessel draining to the axillary lymph node in a rat. There's an example of a mouse tail, and then the top is a mouse tail that's been injured, and so there's basically a cut in that top lymphatic vessel that's dumping out, and then all the surrounding capillaries are picking that fluid back up. So this is a technique we've been working on in our lab, and we wanted to figure out how we could use it to really understand function. So classically, lymphatic function has been determined by measuring diameter. Now with this technique, there was a little bit of a question of, well, can we measure diameter? Because we don't see the vessel, we see the results of the vessel transporting fluorescent fluid. And so what we did a handful of experiments. What we see here is on the left is sort of our classical measurement of vessel diameter, and on the right is that vessel transporting fluorescent fluid. And what we found was that by tracking sort of the intensity of the vessel, we get at least the frequency and some indication of the strengths of the contractions, and that lines up quite well with the actual diameter itself. And just as an example here, at this period, say, of systole, when the vessel is constricted as much as possible, as you can see on the bright field image, we get very little fluorescent signal because there's no fluorescence. The volume of that vessel is very, very low, so the fluorescent signal is really low, versus when it's dilated, or what we call diastole, our fluorescence is maximal. Now, this is all done on a vessel we've taken out, but what happens when we throw skin on top of it, which is ultimately what we do in vivo. So, we get these beautiful, clear images when there's no skin. Now, we throw skin on top of it, and obviously they become very blurred because of a process known as tissue scattering. If you take a laser light pen and sort of shine it through your thumb, right, you won't see a discrete dot. You'll see this nice glow, and that's because the light's getting scattered throughout your tissue as it passes through. But what we found is that we can these regions of sort of large fluorescence basically correspond, and I'll just line this over, with this sinus region of the vessel. And then in between, we have sort of the tubular regions that are smaller, and it's in those regions that we can really see rapid changes in fluorescence. And so then what we can do is things like this. So in this example here, you have this region of interest painted over or selected over one of these tube regions, these narrow regions, and the fluorescence as it goes by spikes up and then goes back down. And we can do this over time, and then we think of, well, can I want to apply an equation to this so I get a number, and I can say, hey, this lymphatic is working 10, this one's working 7. I mean, those aren't the real numbers, but you know what I mean. I want a number that tells me something about this transport. So what we do is we take this curve, we perform an integral of the curve, which is essentially telling us how much fluorescence has passed through this vessel over time, and we apply the little equation to sort of give us a number of total fluorescence transport. And then we'll use this number in some studies I'll show you here in just a minute. So now that we have these tools, what we wanted to do is to go into an animal model of lymphedema, of lymphatic failure, to answer some unique questions. So the mouse tail is probably the most widely used animal lymphedema model. It has a couple of really unique advantages. One, it's relatively low cost. There's a lot of genetic mice available in which I can take, say, mice that have genes that have been implicated in lymphatic deficiencies that are knocked out. I can do experiments there. The tail is a really nice limb, so it's a limb in a sense of it's a sort of a uniform geometry, and it's very easy to capture its swelling. And I think the length scale of the tail is probably actually closer to a human. So, right, a mouse arm only has to transport fluid a centimeter or two, where the tail you actually have a decent distance that fluid has to be transported along the lymphatic chain, similar to like what occurs in a human arm or leg, with a big exception of gravity. All right, but the main thing with these models previously, the way they work is you would make, if you can see right here, that you do an incision, and essentially you cut every single lymphatic vessel, both the capillaries and the pumping vessels that drain the tail. And then when doing so, that tail swells over time. And then there's been a lot of papers that have captured this swelling and sort of what occurs. But if we think about lymphedema and breast cancer-related lymphedema, there are multiple collectors that drain that arm. And during surgery or radiation, some of those collectors may be disrupted, but not all. And there are numerous studies showing, immediately after surgery, you still have patent functional collecting lymphatic vessels draining the arm that were uninjured by the surgery. But now, those vessels have to handle the fluid load of the entire network. So let's say there were 3 vessels and 2 get damaged. Now 1 vessel has to do the work of 3. And so the question we want to understand is, how do the surviving collectors that were completely normal even after the surgery, how do they adapt over time to handle this additional fluid load? So we went back to the mouse tail, and it turns out in the mouse tail that you have sort of 2 chains of lymphatic collectors running on either side of the vessel, which is quite advantageous for us because we can surgically disrupt one, leave the other intact. We can use imaging to ensure we have done both of those things, both surgically disrupted one while at the same time not harming the other vessel at all. And then we can track the function of that vessel over time. So for all this data I'm going to show you after here, it's not data on the vessel that was wounded, rather it's data on the vessel that was unharmed. So this is how this experiment went. We take a baseline measurement of lymphatic function, so we know how well those vessels are working beforehand. So we call it the surgical day. And then we do follow-up measurements of function at various time points after the surgery. And then we can measure tail swelling throughout this experiment as well. Now, one thing I just have to introduce a concept so that you can understand the data. We found in many of the animal limbs we've looked at, in the tail, et cetera, that you often have one vessel that pumps a lot stronger than another. And so we've adapted the convention or the nomenclature referring to this as the dominant vessel versus the non-dominant. So this is an example. In the dominant vessel, usually fluorescence gets there faster, velocity is higher, it pumps more, etc. And so in some of this data, you'll see that we kept track of whether we injured the dominant vessel or the non-dominant vessel. And perhaps it would not be surprising that if you injure the dominant vessel, your swelling is most severe. Again, this vessel seems to be the one that's the most relied upon for lymph transport. And when that vessel is injured, the swelling is the most severe. When you injure the other one, you still have swelling, but it is not as severe. And we do a sham surgical control where we still make incisions and have a wound. We just don't hit, and the dermal lymphatics get damaged a little bit. We do not damage any of the collecting vessels. And again, it really takes the damage of the collecting vessels to get a lymphedema in this model here. All right, so then what we can do is now we can go in and actually look at how function changes over time. So in the sham surgeries, the intact vessel continues to function completely normally over the course of the healing process of that wound. When we damage the non-dominant vessel, the weaker vessel, we see that the other vessel has some loss of function that does eventually sort of get close to being restored 3 months later. But in the other case, when we damage the dominant vessel, that weaker pump is now having to compensate. And you'll notice that, say 2 days after the surgery, you have some reduction, but it gets weaker over time. So this vessel that was intact seems to lose its ability to pump. And this is all using this fluorescence transport metric. Now we could also break that down and look at frequency. So you can think of this as how many contractions per minute it's generating. And we see in this case here that if you wait long enough, the frequency of contractions goes back up. So this vessel is still trying to pump as frequently, but the packet amplitude, which is a measure, if you think about that slide I showed earlier, it's the strength of that contraction. The packet amplitude is significantly impaired. This vessel can no longer contract as strongly as it did, and this is even up out to 3 months afterwards. Now, one way we could plot this data is to actually look at correlations. And so if we correlate, the bottom axis is essentially the swelling, and the top axis is how much that vessel loses function over time, we see a strong negative correlation. So the worse off that vessel responds to the surgery, the higher the swelling. Really providing a strong connection between vessel function of a vessel that was not damaged during the surgery and the resulting pathology. Now, interestingly, in the mouse tail, typically that swelling goes away in all cases. And if we break out and we look at this correlation as the swelling occurs, we see a strong correlation. But if we look at after the fact, the correlation completely breaks down. So we do have restoration of the swelling, but the dysfunctional lymphatic persists. So what this suggests is there's some sort of compensatory mechanism in the mouse that allows the swelling to go back down, even though the lymphatics no longer working as well. And I'll show you suggestions of what we think that might be. Now, one other thing we wanted to develop was a way to measure the pumping pressure. So, blood pressure is a common clinical diagnostic that can be used to sort of tell me the health of my heart. Could we do something similar for the lymphatic? And in this work here, which was published about a year ago, we developed this system to do this in a rodent tail. And basically what it involves is we take a pressure cuff, just similar to a blood pressure cuff. You can kind of barely see the border of it there in this image. And what we do is we allow fluorescence to fill in the vessel, then we inflate the cuff. All the fluorescence empties out. And then we gradually deflate the cuff in a controlled fashion. Again, we use what's called feedback control to do so. And we observe the fluorescence recover as we deflate that cuff. And so what that allows us to do is we can figure out at what pressure we begin to see flow returning in the lymphatic vessel. And that tells us the pumping capacity of the lymphatics on the distal end of the tail. Right? And so we would call this lymphatic pumping pressure. In addition to that, we can also measure the rate of emptying. So we do this occlusion, and we can measure how quickly fluid leaves the vessel after we include. And this tells me essentially the function of the vessels on the proximal end of that cuff. And if we do that, here's an example, right, where we inflate to 80 millimeters of mercury, and at 3 different time points, we see that vessel empty over time, and we can capture the rate at which that empties over time. So we did this at 3 months out, and at 3 months out, our vessel, what we call our dominant vessel, had a completely normal emptying rate. So it can empty fluid at the same rate as sham, suggesting that all those vessels that are on the proximal end of the mouse towards the body, if you will, that those are functioning okay. But when we measure lymphatic pumping pressure, which again, this is just to illustrate it, we figure out what pressure do I begin to see flow return. That actually is significantly impaired at 3 months out. And so while pumping pressure is completely normal a few days after surgery, so these things can pump just fine, if I go far enough out, the vessels on the distal end, so towards the tip of the tail, distal to the wound site, can no longer push fluid past this constriction. All right, and this is further evident by this video here, and what you'll see happening. So on the top is this is several months out. This is, on the top, it's my surgery, and the bottom is my sham wound. And what we see happens as I play this video, so that we've shown there's this underlying deficiency in that pump, right? So the pump is not working as well. So what happens is, the fluid takes the surface streets, right? So the pump doesn't work as well, and so what the fluid does is it just pushes its way through the initial capillary network instead. Remember, those vessels have no muscle, but if the interstitial fluid pressure is high enough, which it certainly is from the injection, fluid can just be pushed through passively. And we can capture this by just capturing the area of fluorescence covered in this tail, and shows that that in fact, in the case of this remodeled vessel, we have significant now reliance on the initial capillary network to try and move fluid versus the collector. And then the last thing we did, and this is going to be my last data slide, is we went in and we looked at these collectors after the fact. And so in the bottom left is my sham surgical control, and this looks like a normal vessel. So we excise these vessels. These have been imaged with a multiphoton microscope, where we can get both the elastin and collagen, which are just sort of materials that make up the vessel. And it looks rather normal. Now if I look, again, these are images of the vessel that was not damaged. So this vessel looks just like sham the day after surgery. But if I wait long enough, on the proximal end, where again our functional data suggested that those vessels worked fine, because remember the emptying rate was normal. The vessel looks relatively normal. But on the distal end, where again our functional metrics would suggest that that vessel is not pumping as well, these vessels are grossly enlarged. And so, and we see tons of capillary lymphangiogenesis. So what we think is important about this suggests that sort of a lymphangiogenic response that would promote lymphatic capillary sprouting and initial lymphatic proliferation is going to have consequences on the collecting vessel in the immediate proximity, and that those responses may not actually be beneficial to its function. So this could be sort of similar to, if we think about the snowstorm analogy, if I clear up all the surface streets, that's only going to do so good. I have to eventually fix the highway so that people can get out of town. And I think this lends evidence that really we need to be thinking about collecting vessels and how to restore that collecting vessel morphology and function in our therapeutic approaches for treating lymphedema. So with that, of course, none of this work would be possible without all the people that do it. And so I just am really fortunate to work with a great team of scientists. I'd particularly like to point out Dr. Mike Weiler, who just recently defended his Ph.D. and has graduated, and he really drove most of this work you see in the lymphedema model and a lot of the initial LE&RN imaging work. Dr. Jana Nipichka does all the isolated vessel work in our lab and molecular mechanisms regulating that contraction. Alex did all the computational modeling. Tyler develops the pumping pressure measurements. And Jeff Cornetta, he also recently graduated, as did Alex, and he developed the vessel system. And two collaborators in particular that contributed to this work. So Dave Zavea, he was actually my PhD advisor and taught me everything I know about lymphatics, as well as he was really integral in helping us set up the isolated vessel procedure in our own lab, and we continue to work with him on that. And then Dr. Reed Gleason, who's at Georgia Tech and does all the computational work with us. And then the two main sources of funding that helped with this work. And with that, I also really like to thank the Lymphatic Educational Research Network. I was very fortunate in beginning a PhD in lymphatics right around the same time that LRF, which is what it was called at the time, was formed. And really I think I was personally benefited by this strong advocacy group that went to Washington and said, hey, this is a disease that's really important that no one understands. We need to fund fundamental research if we're going to understand this. And I was just really lucky that I decided to do a PhD right around the time that all that work was happening. And I know my career has personally benefited from the fact that there is this growing interest in the research community in lymphatics. I know I went through this really fast, and our lab has a lot of other things, so I'll just refer you to our lab website. We try to constantly update that and put videos and things up there, and I'd be happy to take any questions. Question was, is there anything known about the neuromuscular complex driving the lymphatic system? We know some things, although I would say we know very little. Collecting lymphatics are innervated. They do, respond to adrenergic stimuli, for example, acetylcholine, things like that. Now, in the context of when you throw that vessel into the physiology where the mechanics are changing and there's cytokines circulating, how important is the neurological control versus all the other cues? I think that's really an open-ended question. The contraction of these lymphatics is very erratic when you compare it to something like the heart, for example. And at any given time, I hear gravity is driving flow through my arms, but my legs need to pump. And so it's really, you can think of it as like localized hearts everywhere that have to constantly adapt. That's a great question. Any questions coming from the live stream? Yes.
I just wondered how you envision the work in your lab affecting, the patients out there.
Sure. All right. So that question was, how do I envision the work in my lab affecting patients? And that's a great question. It's one I try to pose to my students on a regular basis, because at the end of the day, a Ph.D. student, I showed you a Ph.D. project that boiled down to one slide, and it can feel very narrow in focus as you specialize. And so even when doing so, you need to think about, well, what's the big picture? I think one of the things we're interested in is early diagnostics of lymphatic failure. So, there's a lot of good clinical evidence out there that suggests if you can detect lymphedema early, even like subclinical, and start conservative management, that you can really improve disease outcomes. But that's not a trivial thing to do. It involves education, which certainly LE&RN is helping. But even if everyone's educated, it's still, they don't necessarily have the tools at their disposal to detect their own lymphedema as early as we would like to. So, for example, could lymphatic pumping pressure be a diagnostic that would be predictive of lymphatic failure risk? And could we, adapt that technology and commercialize it in such a way that it actually can be accessible to patients? I think there's ways to go with that. Or can we improve the standard tape measure, approach that we use often throughout the clinics to assess lymphedema? Can we automate that? Can we get it to where it's consistent across clinics where you don't have interoperator variability, all those sort of things? So that'd be one example. Another would be if we can figure out the molecular mechanisms that are driving this weakening of the pump or this unwanted enlargement. If that holds to be true, then maybe that gives us a drug target to stop that from happening locally. And maybe we can preserve the function of those intact lymphatics or do things that lower their risk of failing. All right, so the question from the audience was, in these mouse tail models, we see the restoration of tissue volume, right? The swelling goes back down, even though we have pretty convincing data that the pumping is permanently at least impaired out to 3 months as far as we've gone out. And so the thought is maybe one of the reasons for that because lymphangiogenesis in that case does help. And I think that's a great question. I think probably so. I haven't shown you the data on the wound, but we have a lot of data on the wound site itself, and that does get repaired. So at the end of the day, you do reconnect that blockage. It does not really pump anymore where it's reconnected, but you do sort of open up more surface streets, if you will, to help. And I think that probably, and maybe because of the fact that the tail, the tail has to work very little against gravity. It's almost completely horizontal. It really doesn't take a ton of pressure to push fluid and return it back. I think that's one limitation of all these really small rodent animal models is, we get lymphedema in places that are far away from the thoracic duct, right, where they encounter lots of gravitational load. So head and neck lymphedema patients have problems when they sleep because gravity is no longer driving flow, as would be the case when they're walking around and upright. And so they have more swelling at night, versus leg lymphedema is the opposite, right? During the day, it takes a lot of work to move that fluid up the leg and back into the vessel. So in the context of situations where there is no gravity and you really don't need a ton of pumping, maybe just having more tubes is actually really, really beneficial. Yes, okay, great. So the question is, I'm a bit more familiar with the IC green method for lymphatic imaging. So can I comment on the method with the it's actually near infrared is actually what we use. So they're actually the same. So I think the term IC green is a little bit of a misnomer. I mean, it looks green to the eye if you put it in solution, but its fluorescence is actually not green. Its fluorescence is in the near infrared around, just to give you some numbers, so green would be about 550 nanometers. Red light is around, say, 630 nanometers, 600 nanometers. ICG is out at around 820. And so ICG is a type of near-infrared fluorophore. We actually started our work with using ICG, and what we found is if you do repeat injections, and I'll refer you to, there's a paper on this that we've authored if you're curious. When you do repeat injections over short time intervals, say 3-4 days apart with ICG, the dye itself slows lymphatic transport and enlarges the downstream lymph nodes. So our measurement that we were using to measure lymphatic function was causing lymphatic dysfunction, which made it difficult to sort of capture the changes in dysfunction that would occur over the disease. So we use a commercially available near-infrared dye that's produced by Lycor, and it has about the same fluorescent wavelength properties, and we take that dye and we bind it to some molecule like a PEG, basically something that's large so that it will go into lymphatics, and that's what we use. So all our studies that that I showed you here, we use that dye instead. The challenge is when you want to go in human, ICG is FDA approved, and it's been FDA approved since the 60s, although not for use in lymphatic imaging, but it's easier to sort of, as a physician, to use it off-label in these kind of feasibility studies and clinical trial studies. So it's unclear the regulatory pathway that you would have to overcome to get a new tracer approved for human use. But for all our animal work, since we don't have to worry about FDA regulations, we've used other NIR fluorophores now. But the actual optical properties of them are the same as ICG. They're actually a little bit brighter than ICG. ICG itself is not a very bright fluorophore. That's a very good question. Yeah, in the paper I referred to, if you're interested, Teresa, it's in Trends in Physiology, and Mike Weiler is the first author. But if you go on my website, all the papers are on there, referenced and linked so that you can get them. What's next for my research? That's a good question. So I guess related to what I talked about here, at least, we've been working with a collaborator at the University of Georgia to develop a large animal model of lymphedema in the sheep, similar to what we did in the mouse tail, but where we have gravitational load, and we can get sort of a persistent swelling, lymphatic failure, but at the same time be able to do the functional imaging. So we're working on that. One of the things we're working on in collaboration with Mari Muthachami at Texas A&M is the muscle cell itself and how the muscle cell might fail in the process of this disease pathology. So as this vessel thickens, gets stiffer, you can think of it, the muscle cell is going to feel something different. And at what point does it cease being a lymphatic muscle cell and start to become something else and lose its ability to contract? So that's something we're interested in. And we have some other things that I didn't really get a chance to talk about, role of lymphatics in transporting fat from the intestine. And we're also really interested in the molecules that are causing these very interesting biomechanical responses to the oscillatory flow. At the end of the day, the endothelial cell has to be able to feel that flow and send a message to the muscle cell to say, okay, contract now, okay, dilate now. And we don't know what those messages are, and that's something we're interested in. So can you comment on the relationship between the functional implications of lymphangiogenesis versus large vessel transport work? So at the risk of repeating myself to all these people, I guess first is what I answered to Josh in regards to there may be scenarios where lymphangiogenesis might help restore normal conditions if you don't need a lot of pumping, if there's not a lot of gravity, for example. But in the vascular community, we actually have a word. So we talk about angiogenesis when we talk about capillary growth and sprouting, and then we talk about arteriogenesis. We actually use a different word even when we describe and discuss the growth of new mature blood vessels, and we don't do that in the lymphatic community currently. We sort of just everything's lymphangiogenesis. So maybe Dave Zavea, maybe you can coin the term. I say collectorogenesis or something like that. Lymphangianogenesis, maybe. But I think we really don't understand everything involved in the development and maturation of collecting vessels. And so, how does VEGF-C, for example, play a role in that process? And how does PROX1 play a role in the transition from an initial to a collecting, if any role at all? There is some interesting work by Tatiana Petrova, and that's come out in regards to the triggers that help the valves develop, which is certainly one major feature of the collecting vessel, but we don't know really much at all about the muscle, how it develops, how it matures. So, and we don't know really the consequence of lymphangiogenesis. So if you have all these molecules like VEGF-C that are circulating to enhance lymphangiogenesis, what happens when those bind to VEGFR3 on a collecting vessel? And is that a good thing or a bad thing? Maybe, I don't know, it might depend. I think it's a great question. Okay, how might your work translate with lipolymphedema or lymphedema? So that's an interesting question. One of the things that I really got interested in when I was doing a postdoc with Melody Schwartz was, is all lymphatic transport the same, right? Is everything just sort of dumping in and kind of flowing along the ride with fluid? Or do some things, maybe immune cells, maybe lipid, get transported and taken up differently by lymphatics than sort of stuff that's just washed along by the flow? And I think the fat one is a very interesting one because, all dietary fat is taken up by your lymphatics that drain the intestine and not the blood capillaries. And so, I think there's some, Veronique Angeli in Australia's got some really interesting work, along with Gwen Randolph at Washington University St. Louis, in sort of the role of lymphatics in reverse cholesterol transport. So it seems that most of the tissue being scavenged, most of the fats are being scavenged from the tissue by HDL from adipocytes, returns to the circulation through lymphatics, and lymphatics express transporters, the cholesterol transporters. So there's that sort of possible implication of you could have lymphatic failure of cholesterol transport that's sort of different from general lymphatic failure. Likewise, in the intestine, all those chylomicrons, how they get in. A study I didn't have time to talk about, but Ph.D. student developed a system where we can look at fat transport from the intestine, and one of the things we found is that in the situations where the fat loads in the lymphatic muscles are really high, and this is acutely, right? So this isn't like a obese model. This is just, a quick McDonald's meal, if you will, for the rat. I don't know if I'm going to get in trouble for libel, but when you do that, this rapid infusion of fat into the lymphatic decreases its contraction and causes a strong tonic constriction. We're in the process of publishing that work, but hopefully it'll be available soon. So I think all these ideas of lymphatics transporting lipid in maybe a unique way could have implications in sort of lipo-related lymphedema. Certainly from a purely mechanical standpoint, as all this fat is deposited, that's going to change the mechanical environment and architecture in which those initial capillaries exist. And I don't know, does that compromise their ability to take up fluid if they're anchored to, in a tissue that's basically composed of a bunch of fat cells versus sort of, solid matrix constructs? I think that's a very interesting question too. So we know, radiation, number of nodes dissected, et cetera, all those increase your risk for lymphedema. And one of the only other things that's consistently evident in the clinical literature is that obesity increases your risk of lymphedema. So I think it's a very, I think the lymphatic and lymphedema community is very aware of the obesity issue. I don't think the obese community is very aware of the lymphatic issue, and I think that's really where there could be very interesting kind of research potential.